Hi everyone. I'm just going to follow up yesterday's videos on from the variation and diversity booklet with the next section. So starting on page 5 of that booklet, looking at genetic comparisons using proteins, DNA and RNA. And these genetic comparisons are used as methods for classification, for grouping organisms according to their similarities. So we'll explain how a phylogenetic tree can be generated using these genetic methods. I'll have a bit of a look at how phylogenetic trees were produced in the past using observable measurements and observable characteristics, features of the organisms, and maybe explain why a genetic method is more accurate and precise. We will introduce these words or use these words degeneracy and introns as we go through. Okay, and there's an exam question as well we'll look at. So, in the past, or originally, we might say that observable features in organisms were compared. So, with those observable features, and in fact, with all classification, we're looking for more similarities means that they are more closely related. So, in many phylogenetic studies, then, looking for those similarities in the observable features can be used to find how closely related. Remember, if you remember back to yesterday's lesson as well, closely related means they have a really important phrase, a more recent common can't spell ancestor. Okay, so we've got some this is the classification of bears. We've got these at the top. This is the kingdom. So the kingdom, phylum, class, order, family, genus, and species. Oh, it is actually written there. So with those classification using the observable features, it was possible to identify these bears. I think that is a Arctos. Is that a brown bear or a grizzly? One of the two. I can't quite remember. Anyway, that's fine. But what we've actually found since, or in more recent times, is that with advances in genetic technology, there is more evidence for phylogenetic relationships. And this is the important feature, more evidence. And because of the increase in the evidence, that means that the phylogenetic relationships are more accurate. Okay, as it were, and we've actually found in some cases that some organisms may have been classified according to their observable features, but since using genetic technology, providing more evidence, that we can actually change the phylogenetic trees to make them more accurate, and we'll have a look at an example of that later. So, the three methods by which genetic technologies are used is by looking at a comparison of proteins, and specifically the amino acid sequence, or a comparison of the nucleotide base sequences in DNA and RNA. I'm going to talk about those in a bit more detail. Okay, so the first of those are the amino acid sequence, the DNA base sequence, and lastly is the mRNA base sequence. So let's look at amino acid sequence first. I did briefly touch on this on the previous video, but it's possible to take a common protein, so a protein that exists in different organisms, and to compare it. So, for example, a protein that exists in lots of animals might be hemoglobin. And hemoglobin obviously is made up of it's made up of 4 polypeptides, and by analysing those polypeptides, we're able to determine what the sequence of amino acids are in those polypeptides. By taking the hemoglobin, a specific protein, from 2 different organisms, we can then compare that sequence of amino acids to look for differences. So if we look along here, we can see we've got lots of the same amino acid. Then, in a different species, we can see that this amino acid has changed, okay, because there has been a mutation in the DNA base sequence. So we know that if we've got 2 pieces or 2 polypeptides, and there is an amino acid that is different at some point along that polypeptide, it must be a result of a mutation. Now, mutations are random and spontaneous. Okay, they always happen. And throughout evolutionary history, they have happened. That's what makes organisms different species. When a new species arises from a pre-existing one, we call that speciation, those 2 new species will be very similar to their common ancestor. However, over time, both of those species, both of the animals within those species, will accumulate mutations. So over time, those mutations add up. So the mutations in the DNA will add up and will change the amino acid sequence in a common polypeptide. So by counting or looking for the differences in the amino acid sequence, we are able to determine firstly that those 2 organisms, 2 individuals from the different species are in fact the different species, but we're also able to add a measurement of time and how long it took for those mutations to occur. So this down here is a question about analysing an amino acid sequence of a blood protein, such as hemoglobin, from hummingbirds. So this is the question: explain how these sequences could provide evidence as to whether the crimson topaz hummingbird and the fiery topaz hummingbird are a different species. So going back to what we just said earlier on, the different species would have different amino acid sequences. The reason for that, for the second mark, this is a 2-mark answer. Amino acid sequence is the result of the DNA base sequence. Okay, so closely related organisms will have more similar amino acid sequences. Organisms with a more distant common ancestor will have more different amino acid sequences. Okay, so closely related, we remember, means a recent common ancestor. 2 species that are not closely related will have a more distant common ancestor. Okay, so hemoglobin could be used, but there are other proteins, and quite a common one for comparison is a protein called Cytochrome C. Cytochrome C is a coenzyme involved in aerobic respiration, and it's been conserved. It's very highly conserved, and that means that it remains in lots of different species from a very distant common ancestor. So what we have here are different branches of eukaryotes. Okay, so cytochrome C is common amongst all eukaryotes, but the specific amino acid sequence in that cytochrome C may change. So down here we have fungi. Here we have, this is the branch of animals, but here we have invertebrates, fish, birds, and mammals. And all of those organisms contain a cytochrome C protein. But the differences between those cytochrome C proteins is due to the accumulation of mutations over time. So there was a common ancestor down here. That was the common ancestor of all of those eukaryotes that we're looking at here. Now, the length of these lines represents two things. So this is the annotation from the diagram. So values are an estimate of the minimum number of nucleotide substitutions that have occurred. So it's an estimate of the mutations, okay, or the minimum number of mutations that must have occurred to separate out each of these different branches. Now, we know that mutations happen at a roughly constant rate. So we can also assume that if they represent the number of mutations, they also represent time. Okay, and there it's possible to make an estimate, therefore, of how far back in history that common ancestor for mammals, birds, fish, invertebrates, and fungi exist. Okay, by looking at the differences in the amino acid sequence. Okay, so you've got this table in your notes. You can see it here. Cytochrome C is a protein. The table shows the sequence of the last 6 amino acids in cytochrome C in humans and 3 other animals. So we are comparing the cytochrome C in humans to monkeys, or a particular species of monkey, a particular fish, and a horse. Now, if you count the differences in the amino acids, we can assume those differences are due to mutations. Okay, or they are due to mutations. So more mutations means a more distant, or more difference, more different amino acid sequences means a more distant common ancestor. And more similarities, vice versa, means a more recent common ancestor. So, if we look, the human and this has the same amino acid sequence. Okay, so we can assume, therefore, that this one is the monkey. Okay, we know that the horses are also mammals, so we would assume that whilst they are quite different to human, but they are more similar. There are fewer differences in their amino acid sequence. There's a horse and a fish, which are a different branch of, or a different kingdom, have more differences in their amino acid sequences. Okay, so that is just the last 6 amino acids. This is a much larger comparison of lots of different species. And where we see that the colours remain the same, where they are conserved, that means they are obviously the same in these different species. We've got humans at the top, rhesus monkeys, what's that? A horse, donkey, cow, pig, sheep, etc. So by comparing the amino acids at each different point along the polypeptide, we're able to determine just how closely related each species is, and therefore how recent their common ancestor is. And using a comparison like that for all for eukaryotes, we can compile this phylogenetic tree like such, like so. Okay, yeah, these ones down the bottom are plants. These are different types of fungi. I can't quite read that. Oh, invertebrates. Birds, I think. Okay, so we can see the differences between those, and that enables us to produce this phylogenetic tree. Okay, so that is comparing amino acid sequences. Now, obviously, the amino acid sequence is determined by the DNA base sequence. So we can use a DNA base sequence for comparison as well. So by comparing DNA that codes for a particular protein, again, when we're comparing the DNA, we need to know that protein. So that protein needs to be one that is conserved or is common, is present in each of the organisms that we are trying to compare. So present in the organism. Okay. And it's possible now with automated techniques and modern sequencing techniques to actually compare whole genomes of organisms. They probably predicted that this is much more accurate and a much more precise way of looking for differences. The reason for that is because of the genetic code. So we know that the genetic code, firstly, is degenerate. It shows degeneracy. That means more than one triplet could code for the same amino acid. So that might explain why that the human and the monkey here appear to have the same amino acid sequence, and it would be difficult to distinguish between them. Now, it could be that those amino acids, whilst they are the same, that the DNA code is different. It just codes for the same amino acid, okay, due to that degeneracy. So by looking at the DNA base sequence, we ruled out the differences that might be hidden by the degeneracy. It also includes non-coding DNA called the introns. And those introns, they differ between different species. So in fact, in some cases, the proteins produced may be very similar amino acid sequences. And we can see that at the top. We can see the rhesus monkey and the human and the chimpanzee. In fact, the human and the chimpanzee, there are no differences all the way across. However, the introns, those non-coding regions of DNA, present only in eukaryotes, those non-coding regions of DNA differ a lot, and we actually see a lot more variation in the introns than we do in the exons. Okay, so by comparing the DNA-based sequence, it's perhaps it is a more accurate, it gives you more evidence to look for comparisons. So again, this idea is really important. More closely related, i.e. they have a more recent common ancestor. I'm going to keep repeating this because I can't emphasize how important it is. More recent common ancestor. Organisms have more similar DNA-based sequences. And we need to use that phrase, DNA-based sequences. Okie doke. So let's have a look at an example of that. Okay, these are 3 species of python. This is olive python, Maclotti python, and the Papuan python. Okay, now they are classified here in 2 different ways. We can see 2 different phylogenetic trees. Now, classification X, this one here, was an earlier classification just using the frequency of observable characteristics. So I looked at the skeleton, the morphology, and a number of different observable characteristics, and looked at how many times they appeared in each of these pythons. And it was determined then that the Liasis Maclotti and the Liasis olivaceus, that's the one. Oh, terrible with reading out species names. Were more closely related. They have a common ancestor that is more recent than the. Sorry, my dog's just come in. More recent than the Papuan. Than the Papuan python. Okay, so there is the common ancestor. Oops, I got distracted by my dog. There is the common ancestor of all 3 down here, and and that is more distant than these 2. These 2 appear to be more closely related. But in fact, since comparisons of genetic characteristics, so there's other genetic characteristics are used, the classification of the python family has changed. So we can see that the Papuana and the Maclotti here are actually the more closely related because they have a more recent common ancestor, and the olivaceus has a common ancestor that is more distant. Okay, so this demonstrates the idea of classifications changing as more evidence becomes available. And in fact, the best evidence available is are the genetic comparisons. Okay, right, so there's a question on this. This is an old exam question. What do these classifications suggest about the evolutionary relationships? Well, we've already mentioned this. The Maclotti, in classification X, the Maclotti and olivaceus have a more recent common ancestor with each other than the Papuana or the Papuan python. Classification Y, this part here, classification Y, well, we can see that the Papuana and the Maclotti have a more recent common ancestor than those two and the olivaceous. Okay, so that was I think that was a 2-mark question, and just using those phrases, more recent, more distant common ancestors, we were able to compare the 2 phylogenetic trees. So state 3 comparisons of genetic diversity that the scientists used in order to generate classification Y. Well, we already know that they could, the most accurate way would be to compare the DNA base sequence. There's one. Second one we've mentioned already is the amino acid sequence. I'm using the abbreviation here for amino acids. Obviously in the exam it's important that you write amino acids. I'm just being a little bit lazy. The third comparison that we've not spoken about yet, which is in your notes, is a comparison of the mRNA base sequence. Okay, and we'll look at that next. Similar to comparing the DNA. However, there are some useful and some perhaps slightly less useful features of comparing mRNA. Isolating mRNA from cells is often much easier than isolating the DNA. There may be only 1, perhaps 2 copies of the DNA, whereas if that mRNA is being transcribed, or if that gene is being transcribed, so maybe only 1 or 2 copies of the gene, but if the gene is being transcribed, there may be hundreds, perhaps thousands of copies of the mRNA. Okay, and that idea about isolating mRNA rather than DNA, it comes up again later next year when we look at gene technology, DNA technology, and the idea that extracting mRNA is easier because there are many copies. It's also easier because it's in the cytoplasm rather than the nucleus. And, well, perhaps this is a bit of a drawback. It consists of the exons only. So the mature mRNA has undergone splicing. So the introns have been removed and now it contains only the exons. So that's perhaps a drawback because, as I said, those introns show lots of genetic variation, lots of variability that might not be present in the exons. So there's a bit of you need to make a decision about whether the ease of isolating the mRNA compensates for the reduction in the precision, perhaps, of using mRNA. The final factor about this, which might be a bit of a disadvantage to using the mRNA, is it only considers genes which are what we refer to as being switched on. So we know some cells switch off certain genes if it's not necessary to make that particular protein in that particular cell, and that's what we mean by in differentiation. In differentiation of cells, so all stem cells in an embryo, in a very early embryo, are identical, all expressing exactly the same genes. As those cells become differentiated, certain genes get switched off, others remained switched on. mRNA will only be produced in those genes which are switched on. Okay, so that's something to consider. Okay, so that's everything really on the methods of classification. We talked about the usefulness of those 3 methods at the end, and there's a bit more detail in the notes, but before my dog starts barking at me, I'm going to sign off for now. Email me if you've got any questions, or you can leave a comment below. Right, thanks a lot. Take care. Bye-bye. Dilly, shut up.